Sean Carroll. A bit about Sean's history. He started his academic career at Washington University in St. Louis, where he earned a BA in biology, and then he completed his PhD in immunology at Tufts Medical School. And the list doesn't stop there. So Sean also holds honorary degree, Doctor of Science degrees from the University of Minnesota, as well as Tufts University. He spent many years of his research career as professor of genetics and molecular biology at the University of Wisconsin, where he is now professor emeritus. And as some of you may know, while he was there, he served as the doctoral advisor of our very own Tricia Witt, placing several of us here on one of the many branches of his academic family tree. Not surprising then, Sean's research falls within the vast realm of Evo-Devo, where he explores the genes that control animal body patterns and that lead to the vast amount of animal diversity that we see today. In addition to being an internationally recognized and multi-award winning scientist, Sean is also a prominent science communicator through his efforts in writing, teaching, and creating documentary films, just to name a few. He's currently leading the Department of Science Education of the Howard Hughes Medical Institute, which is the largest private supporter of science education activities in the U.S. And he's also the executive director of HHMI, Tangled Bank Studios, and is the Andrew and Mary Bailo and Nicholas and Susan Simon Endowed Chair of Biology at the University of Maryland. Okay. Really, he's quite an inspiration. And furthermore, I think all of us who have some degree of familiarity with Sean can agree with a few words that Tricia said about him, that he's undeniably very successful and takes his science very seriously, while simultaneously being one of the most down-to-earth people you could meet, who strives to advocate that science is fun, which I believe is so important. I don't want to talk too much, so to wrap it up, I just want to remind you all that Sean will be giving a public talk tonight at 7 p.m. in room 1060 BSB. And without further ado, I'll hand it over to Sean, who will be talking about butterfly spots and rattlesnake tails.
Thanks, Molly. So I'm quite sure that the best contribution I'm going to give to the University of Michigan is not today, but it's having not derailed the meteoric career of Tricia Witt. So, who left just a few days ago for Germany, and then I'm headed to Heidelberg in October, and she told me she's leaving a few days before I get there too. So read into it what you like. So anyway, so thank you again, Molly. Thanks to the students. Thanks for the donuts. Thanks for the chocolate eclair also this morning. So I'm a little revved on chocolate at the moment, but I'm always revved to talk about biology. So today I'm going to talk a little bit about the ideas that have driven my lab for probably more than 20 years. But before I get into the science, I want to just take a couple of moments to think about our motivations and aspirations as biologists, because that played a big part in what I chose to study as a biologist and even what we're studying now. And I think that one of my old friends, Albert Camus, put it best when he said, okay, somewhat chauvinistically, a man's work is nothing but this slow trek to rediscover through the detours of art those 2 or 3 great and simple images in whose presence his heart first opened. I think this can also be true of science, and I've even spoken to people today who I think this is true, that those first glimpses, first experiences can mean so much. For me, those few images were these. On the left is a speckled king snake. One of my first pets. On the right is the Sonoran mountain king snake, to a kid that grew up in Toledo, Ohio, 50 miles from here, an almost mythical creature who I've never seen in the wild. But it was these creatures that launched, and as it turns out, they're spots and stripes, that launched my interest in biology. And Molly gave a resume there, but I've been incredibly lucky to pursue the things that interest me. And I'm aware that for many people that's not the case. So if audio works well on this one, listen carefully.
Well, Mama don't allow no skiffle down in here. Oh, no, she don't. Mama don't allow no skiffle down in here. Well, we don't care what Mama don't allow. Gotta play that skiffle anyhow. Mama don't allow no skiffle down in here.
We were a little local band who were playing around school dances, and not even the slightest, mildest consideration that that would go into something. There wasn't this driving thing that the minute you hit your teens, you've got to be famous.
What are your two names? Yours is?
James Page and...
David Haskell. And you're just learning to play the guitar?
Yes,
Can you from a teacher? Do you play anything except skiffle?
Yes, Spanish and dance.
Do you as well? Can you move on? What are you going to do when you leave school? Take up skiffle?
No, I want to do, biological research.
But young James Page, he didn't get the chance to do biological research. He had to find an alternative career as the lead guitarist for Led Zeppelin. Couldn't even afford a shirt with buttons. So, not Jimmy Page, it's we folks. We biologists, we are living the dream, people. We are living the dream. And my dream, since I grew up catching snakes in the fields and woods around Toledo, Ohio, was to someday study those creatures. I had to make do for a while studying smaller things indoors. So I'm going to talk a little bit about those smaller things before we get back to those slithering creatures. And the question that has guided my lab for certainly about the last 20 years is the origins of biological novelty, a big question in evolutionary science, and that's manifest in so many ways with the creatures that we love in the biosphere. And today I'm going to talk about two dimensions of investigation into the origins of novelty, coming at it both from a genetic perspective. And the first is the origin of new morphologies, and for that's more historical work from my times at Wisconsin. And then the origins of protein functions, new protein functions. I'm going to talk about all new work and the sort of things that are ongoing at Maryland. I want to back up a little bit about what biologists were thinking for a long time before we entered this arena. And the most influential, I think, single piece of work was done by Susumu Ono, who wrote a book in 1970 called Evolution by Gene Duplication. There are lots of folks interested in the dimension of this work here in Michigan, and I'm sure they're quite familiar with the book. But it was a marvelous short book, I think less than 100 pages, that stimulated a lot of thinking and I think framed a lot of thinking for decades to come. I want to just highlight a couple things that Ono said right in the preface, that natural selection merely modified while redundancy created, and that allelic mutations of already existing gene loci cannot account for major changes in evolution. So Ono's view was that gene duplication was a necessary prerequisite to the origins of novelty. And two other great Japanese evolutionary biologists sort of doubled down on Ono's point of view a few years later, Kimura and Ota, by saying that gene duplication must always precede the emergence of a gene having a new function. So that was the mindset. Certainly when I arrived in developmental biology and we're starting to discover some of the genes that were involved in the shaping of body form, our expectations were that we would find genes that would be restricted to particular lineages with different body plans. You may know that that's not exactly what we found, but I'll tell you what we did find and how it led us along another path. In the early 1990s, we started trying to explore the making of diverse patterns in animals from a developmental point of view, and started working our work, for example, in butterflies. And one day in the lab, we made essentially this discovery, which really didn't know it at the time, was going to be pretty much foreshadow a lot of what we were going to learn from the evolution of development. And that was, we were studying a gene, I'll tell you about in a second, that we had known from model species like Drosophila. We were studying its deployment in a butterfly. On the left are immunofluorescence micrographs of this gene in a monarch butterfly and in an East African butterfly. And what you can see in the differences in expression, the gene is expressed along the distal part of the whole wing here, but it's upregulated in these discrete little spots here. And those correspond beautifully to the centers of these spots in this butterfly, which has these eye spots which are used in predator avoidance, while the monarch does not. So we had a clear difference between these two species, 7 new spots of gene use in this species relative to this other species. Just a little morphing, a little circa 1990s morphing to show you that those green spots were going to be the white pupil centers of these eye spots. And that was incredibly exciting, but what was more exciting to us was the identity of this gene, because we were studying this gene for a variety of reasons, It had been identified first in Drosophila. Is everybody okay with the lights out front? Is that washing the slides out, or you're all okay? You're all okay. Good? I heard all the way from the back. Everybody good in the back? Thumbs up from the back. Okay. So the identity of the gene we are studying for a variety of reasons. It had been identified, as I said, first in fruit flies, but we were discovering, particularly Grace Bolkoff-Falk in my lab was discovering that it was used in all sorts of taxa. And the gene's named distalis. And it's called that because in fruit flies, the distal parts of the appendages don't form in mutants of this gene. But in other taxa, it showed a pattern that was really striking in terms of its deployment in different kinds of appendages. So this is a fruit fly, and it's developing mouth parts and imaginal legs. This is a developing butterfly. There's the thoracic legs. There's the abdominal legs, a developing brine shrimp. Its various swimming appendages and other sorts of mouth parts. A developing sea urchin and its tube feet. A marine annelid and its parapodia. Developing chicken and its limb bud. And developing fish fin bud. So all sorts of appendages, all sorts of projections from the body wall were deploying this distalis gene. And so we're impressed that this gene has been around for 500 million years or so, but in lineage of butterflies that make spots, it was being used in a new way. It had taken on a new job. And this turned out to be a theme that we and many other folks were going to encounter in the coming years. And I like rules, and if you come to both talks today, first of all, you get extra mileage from United or something like that. But you'll also see that I'll be talking about different kinds of rules tonight. But I think the first rule from Evo Devo was that new patterns evolve when old genes learn new tricks, that these genes have been around for a very long time. In fact, in their coding regions have been around in their very similar form for a very long time, but can still pick up new jobs in various lineages. And this, as I said, foreshadowed what would be repeatedly discovered in Evo Devo, and gave us a hint that these genes that were involved in regulating the formation and patterning of body parts were different than the kinds of genes we had been studying in, for example, metazoans up to that time. And they were different in that they were pleiotropic. And what I mean by that is there were multiple functions of individual genes. And there was really no expectation, no prediction of that discovery. When people thought about the making of anatomy, I guess people sort of imagined that you'd have genes for making finger bones and you'd have genes for making rib bones, and you wouldn't necessarily use the same genes for making different parts of anatomy of the body. But in fact, not only do you have the same genes for doing things like that, but even the same gene can be involved in really different sorts of things. Let me give you an example of a famous gene. This is sonic hedgehog. In expression in 2 stages of the chicken. And all you're seeing is a view where you can see the developing limb buds. And the dark pattern here is where the sonic hedgehog transcript is expressed. And then here along the developing neural tube. And then a bit later in development in feather bud formation. Okay, so for all the embryologists, developmental biologists in the audience, what do all 3 of these structures have in common? Correct, nothing at all, okay? So there is absolutely no reason whatsoever you would imagine that this gene would be used in making feather buds and neural tubes and limb buds altogether, as well as many other things. So this is what I mean by pleiotropy, one gene with multiple functions. It's the exact same protein form that's involved in these developmental processes in these different sorts of tissues. And the discovery of this kind of pleiotropy, we thought, had some obvious, to us anyway, evolutionary implications. And those implications were that individual regulatory genes have acquired perhaps many new roles in the course of evolution without duplication. So these were single copy genes, something like sonic hedgehog has paralogs, but we're talking about the jobs of sonic hedgehog itself. I don't know, we probably know a dozen or 15 different things that sonic hedgehog does in the making of a vertebrate. In fruit flies, we are finding this again and again, genes playing multiple roles in building embryonic body parts, larval body parts, adult body parts, et cetera. And it means that gene cooption must be widespread. And just the functional definition of cooption is the use of the same thing in new roles. And that could be a gene, that could even be a body part. But I'm really talking about the use of a gene in new roles. But that was great to sort of get this far, but we really want to understand how cooption occurred. And that was really challenging. It just turns out we were not well positioned in studying, comparing things across vast taxonomic scales to really pinpoint the genetic origin of a new piece of morphology or a new piece of pattern. And so from very broad scale comparisons that we were doing, so for example, butterflies to fruit flies or whatever, we returned to sort of our roots and started studying novelties in fruit flies. And this was just at the time that Tricia Witt was in the lab, and her pioneering work on pigmentation genes and gene regulation and first work on the evolution of pigmentation in the lab got us into this business. And so I'm going to just give you just the highlights and I think no data from work that is now quite a bit older. But nonetheless, for us, I think that the challenge in this business, I was just talking with students a little bit, the art in biology, I think, is to find the simplest example of the phenomenon you want to understand. And in our case, the simplest example would be two Drosophila species that differ simply in the presence of a pattern element, in this case, this pigmentation at the distal part of the wing. And we know that this pattern element is involved in a display that males make in front of females, so we think it's behaviorally interesting, but we just treat this as a piece of morphology. And Tricia, in her groundbreaking work, and two postdocs in the lab at the time, Nicolas Gompel and Benjamin Prudhomme, took on this challenge. I'm going to kind of give you the summary figure and then one more little kick. So how does cooption occur? And the picture that we built up from various lines of evidence, I'm not going to show you, is how an existing enhancer or cis-regulatory element and existing transcription factors can be connected, can come together in a new way and create and generate a new piece of pattern. And what that requires is regulatory elements for a gene involved in the making of that pattern element. This is a gene called yellow, should be familiar to most people in biology at Michigan, thanks to Tricia. Think of it as a sort of a paintbrush for making pigmentation, for making dark pigmentation in fruit flies. And if you imagine sort of an invisible landscape in these wings of regulatory factors that are involved in making the morphogenesis of the wing, sort of the macro architecture of the wing, shown here in green and purple, we're going to bring those together with the regulatory elements of this pigmentation gene to generate a new pattern. How does that happen? Well, you have a pretty simple transcription unit here of the yellow gene, but there's a regulatory element of the yellow gene that happens to pick up binding sites for these transcription factors. And these binding sites for these transcription factors are pretty simple. It's easy for them, new sites to emerge through simple point mutations. And if you imagine that this ancestral regulatory element drove a low level of expression of this gene across the entire wing surface, by gaining binding sites for these transcription factors, this activity could be modulated. In particular, if the regulatory factor shown here in pink is an activator of transcription, and the regulatory factor shown here in green is a repressor of transcription, that combined action of those two will be to generate higher levels of expression of this gene in the posterior quadrant, distal quadrant of this wing. So that's an activator and a repressor. And Nicolas and Benjamin were able to show that, in fact, the repressor of this activity was a well-known Drosophila gene, in fact, a well-known insect gene called engrailed, a homeodomain transcription factor expressed in the posterior compartment, not only of fruit fly wings, but of all insect and arthropod segments. So again, this is an old gene that's been around for a very long time that has been recruited into a little piece of morphogenesis that is unique to this spotted fly lineage. For the longest time, so Nicolas and Benjamin left the lab, set up a joint lab in Marseille, France, and we did not know the identity of the activator. So we knew the repressor was the thing in green, but we didn't know the identity of this activator until published, I think, in 2013. And of all things, the activator in fruit flies is distalis. Something almost a religious experience for me, I think. Almost. And you'd think, well, why didn't you look at that earlier? That's a long story. You can ask me some other time. They did look. We kind of got thrown off the trail. But it turns out that trail eventually led them in France back to distalis, and that distalis is modulated in these spotted species in a pattern that nicely preconfigures where the spot's going to form, and it's expressed in a different way than it is in melanogaster. But anyway, distalis is the activator. So There's no change, no functional change to the coding regions of these transcription factors. Binding sites for the transcription factors evolve in these regulatory elements, modulate the spatial expression of these spatial activities, regulatory elements, get a new pattern of pigmentation, gene expression, get a new pattern. So we think that's how cooption occurs, but we also think that's in general an insight into how the development of form evolves, how the evolution of new patterns can evolve. So we think it's a second rule emerging from Evo-Devo in general, that new gene expression patterns and new morphologies evolve largely through changes in enhancers, that it's the regulatory elements that are evolving, and it's the rewiring of regulatory networks, the rewiring of circuits, of interactions between existing transcription factors and existing genes that's generating diversity of pattern. So it's not a story of gene duplication. In fact, sort of in parallel to this work, as we studied the genomes of all sorts of arthropods and vertebrates and other species, it's a pretty clear pattern. It's the toolkit, the genes involved, like engrailed, like distalis, in building bodies and body parts, have been incredibly stable through evolutionary time. And I just want to show you just a sample picture of this, because I'm going to contrast it with what I'm going to show you in the second half of the talk. So if you just look at things like a coelacanth in humans and mice and things like Xenopus, and imagine what was present in the last common ancestor of all these groups, there's no new Hox genes have evolved in 400 million years of vertebrate evolution. It's an incredibly stable pattern. So we don't, and the same story goes on for arthropods. And there's really been no significant evolution of the genes that are involved in specifying pattern along the anterior-posterior axis from Onychophora through the arthropods. So the toolkit is very old and stable. I just want you to put this picture in your mind and contrast it with what I'm about to show you. Okay, so after a couple exciting decades in fruit flies and butterfly land, the old guy decided we had to finally ask some questions about some other creatures. So I'm sure some of you, when you see this creature, you sensibly think, hey, that's a dangerous animal. I don't want to get anywhere near it. But I see something different. I see an evolutionary mystery, or actually a set of evolutionary mysteries, a lot of morphological novelty here. Limbless body, the rattle, forked tongue, the infrared sensing pits that the rattlesnake uses. And actually, when it opens its mouth, it kind of just like Donald Trump, gets even scarier. Fangs and venom. It's also fitting. So a lot of novelty here, but we wanted to zero in this case, particularly on the evolution of biochemical novelties and on venom. And you might ask, well, why snake venom? Well, actually, there's a lot of animals that make venoms, and they'd all be reasonable choices for studying the evolution of novelty. But we thought snake venoms, there's been a lot of interest in snake venoms. Because they're large animals, the venoms are, when I say easily obtained, I've never milked a rattlesnake, but it's easy for other people to do. But you can get copious quantities of venom, but of course there's medical interest in these animals, which has led to a large literature, a lot of study of snakes across the world for the last few decades. But there's certainly, venoms are a key trait involved, of course, in subduing and acquiring prey. They're complex mixtures of proteins, so that sort of sets up some interesting dynamics of how you have sort of you have a novel concoction, in fact, almost an arsenal of proteins. And we knew from a fair amount, from a lot of natural history, that there is both diversity between and within species, and we're going to explore both those dimensions in the course of the next 20 minutes or so. So we thought if we're going to get into snake venoms, we need to start somewhere. And the rattlesnakes, the reason we want to study rattlesnakes, not only is all this true of rattlesnakes, but they're readily available here, and we've had great help from collaborators. Oops, we jumped hard. This skipped a slide. Okay, no, sorry, never mind. So we're going to study the novelty in terms of the origin of protein functions, and generally frame, although I'm not going to answer for you this question in great detail. You'll see little glimpses of this, but in general, we're trying to understand to what degree are venom toxins sort of old proteins with a new job, physiological proteins that are being used in their present form, but if expressed in large quantities or injected into the bloodstream of an animal, can do some damage, or are they new proteins? with new activities. And to get into this work, we needed a lot of help because we had no experience with it. And Elda Sanchez, who's at the Natural Toxins Research Center at Texas A&M Kingsville, has been a great help hosting people from my lab. And so Elda and Mark Huckmuller, who are, that's Elda, that's Mark, and that's Noah Dowell from my lab and Matt Giorgiani from my lab, really helped us get going and learn about these animals and acquire original materials. And Elda and Mark have a deep background in the study of these rattlesnakes. So we got these materials and started going. So let me introduce you a little bit to rattlesnake venoms, just to keep in mind, especially for today's talk, that a lot of the rattlesnakes, in fact, ones that you might be familiar with or have heard of, certainly the big, large-bodied, sort of more famous ones, their venoms are hemorrhagic, but there are also some species that are neurotoxic. And that's what drew us in here. We thought, okay, well, if there's really just venoms acting in distinct ways, we'd like to understand how did some of that activity evolve. So for example, it's the minority of rattlesnakes that are neurotoxic. How did neurotoxicity evolve? So we took a genomic approach to the origins of these toxins, and this, strangely enough, was lacking from the field. There was a fair amount of work on proteins, a little bit of work on cDNAs of genes, et cetera. But we thought if we were going to understand really where these genes came from, we needed to trace their genomic origins. And that's one sentence, but of course, anyone here who's done vertebrate genomics appreciates that there's a lot more to it than that. So I'm going to tell you some of the discoveries that have come from pursuing the genomic origins of these toxins, and we're going to look at two groups of toxins. The first are metalloproteases. These are zinc-dependent enzymes that are involved in causing hemorrhage because they destroy the integrity of the vasculature. I'm going to save you a fair amount of preamble and just give you a quick glimpse to what, in a western diamondback rattlesnake, what the organization of the genes for these metalloproteases looks like, and then tell you a little bit more about how we've traced their origin. And sorry, I'm giggling. There are 30 genes, and the reason why I'm laughing, I think this is half a megabase. This was more than we expected. From proteomic studies, et cetera, maybe we were expecting 10 or 11 at most, but there were 30, and all tandemly duplicated, all tandem duplicates right next to each other. I'm just going to give you a little glimpse. Don't have to worry about the nomenclature too much, but I'm a little glimpse about the genesis of these genes, because it's, again, something a little different than what we were thinking about. And so these venom metalloproteases have been well studied in the snake field, but metalloproteases in general have been studied because there's a lot of physiological interest in metalloproteases, and they're involved in cancer metastases and things like this. So what we know about these genes is that the expansion of this family has occurred through a process of both duplication and deletion. I'll show you the evidence, but it turns out that in this complex of genes I just showed you, there's a metalloproteinase gene that has a transmembrane segment, and that's a lot like metalloproteinases you find doing physiological roles throughout the body. But all of the metalloproteinases expressed in the venom lack this transmembrane domain, and we know that that form of metalloproteinases was generated by deletion of the last, say, 8 or so exons of this gene, several of which encode the transmembrane domain. So in this case, novelty has come, at least one step in novelty was trimming off the part of the gene that encodes the transmembrane domain, and now you have a soluble secreted form of the enzyme. There are 3 recognized structural domains to these metalloproteinases. The metalloproteinase domain, which has the active site, a disintegrin domain, which is involved, I think, in interacting with vascular surfaces, and then a cysteine-rich domain, which is not well understood. And there are 3 types of metalloproteinases, dubbed amazingly 1, 2, and 3. So 3 has these 3 domains together, no transmembrane domain. But there's also type 2s that now lack both the transmembrane domain and the cysteine-rich domain. And in the western diamondback rattlesnake, there's one type 1, which lacks that domain, and that domain. So all of these evolved, and we can map these things very precisely. All of these different classes evolved by deletion from a larger, essentially 4-part protein. And these are not independent events. The type 1 evolved from a type 2. So this was a progressive process in generating the type 1. So, but when we look back in time and ask, well, what's the history of this family? What we see is a massive expansion in genes in rattlesnakes from an ancestral metalloproteinase locus. So if you look at an outgroup of something like a king cobra, something that's not a hemorrhagic snake, or say, for example, boa that's not venomous at all, we can see that all of these metalloproteinases evolve from one or at most two paralogous genes in non-crotalid snakes, non-vipers, into this group of 29 to 30 genes that's there in a western diamondback. So massive expansion. And this expansion, probably less than 30 million years. So this is a really, for my lab's experience, a really different story. That's why I wanted you to look at that Hawks picture. 400 million years of stability versus massive and rapid expansion. And in fact, by surveying just a small number of individuals from the field, we found copy number variation in this species, both here and another gene complex I told you about. So it looks like it's even a fairly labile number or variable number in the wild right now today. All right, let me pivot to another set of genes, a little different, similar sort of take-home story, different job for these genes, but this is where we thought we were going to essentially trace the origin of a novelty within rattlesnakes. And this is the origin of its neurotoxin, which is a heterodimeric protein made up from two phospholipase A2 chains. It's a presynaptic neurotoxin. And we went into one of these neurotoxic rattlesnakes called the Mojave Rattler from southwestern United States, and we looked at the organization of the genes of this toxin group. So you're going to see a lot of these little schematics involving these genes. You're probably going to get a little tired of them, or maybe you don't even care already. But just in case you care about the data, I'm going to give you a little bit of orientation to what's here. So these phospholipase A2s used in these rattlesnakes are all related to each other, and I'll show you in a second, all descended from one gene. But they're neighbored by other phospholipase A2 genes that have not become involved in venom. So it's a multi-gene family from which one family member gave rise to the stuff that's of interest in terms of what's in venom. So these are the not relevant to venom PLA2s right out there. These are the immediately adjacent genes. Everything I'm showing you in color is snake specific. Okay? And these two genes, circled here in the orange and green, encode the two subunits of the neurotoxin. All right, so they're telling, that's where the neurotoxins come from. Don't worry about the other ones for the moment. We really want to know where these neurotoxins came from. So as I was just telling you in a second, we know that all of these rattlesnake PLA2s, all these have evolved from one snake-specific ancestral gene and expanded out. So it's a fairly similar picture to what I just showed you for the metalloproteinases. Not as massive an expansion, but generally an expansion from a single locus in this lineage that gave rise to rattlesnakes. So given that this happened in this lineage, we thought, aha, here we are, let's study this novelty. And given that we knew that some rattlesnakes are neurotoxic and some rattlesnakes are not, we thought, let's trace the origin of this nasty little neurotoxin. Except for, I forgot what I wanted to bring you, why I told you all that. Hold that thought. Just to go back to the first part of the talk and draw the comparison I wanted, there's a reason why I want to talk about massive expansion versus that stability. And this is, I'm just going to put this forth, and maybe through the rest of the afternoon, some of the folks I'm meeting with, we can have a discussion about it. But I do think there's something informative here about the really striking contrast between the history of things like Hox genes and the history of these venom toxin genes. And I think there's a way to rationalize that difference. We see that among regulatory genes, like the Hox genes, duplications are rarely fixed in animals. We certainly think that the gene duplication rate, the forward rate, is going to be the same. That's random across the genome. But those duplications rarely are fixed. But I think there's a reason for that. And there's empirical evidence from developmental biology why this would be true. When that gene product is wired into multiple regulatory circuits, regulatory networks, those networks are sensitive to dosage. And that simply a change in copy number from 2 to 3 copies can have deleterious effects. So we think that some of these duplications, which will have expression on other genes, are actually selected against. It's the best way we can rationalize the lack of fixation of duplicates in these regulatory genes over vast periods of time. In contrast, sort of the pattern of gene duplication we're all more used to from lots of our favorite gene families, duplications could be numerous. We see no reason why they would be constrained. They have no effect on the expression of other genes. These venom proteins are free to do whatever they're going to do. And the duplicates may well be selected for as these genes I'm talking about, for example, probably get engaged in some kind of arms race between predator and prey. So a real contrast in the evolutionary pattern of diversification of these gene families. And it's this component that I don't think has been talked about enough in the literature of why would you not see these duplicates. But it really has to do with the fact that they're wired, I believe, that they're wired into lots of regulatory networks, and that change in dosage will be deleterious in one or more significant ways. Okay, now back to the question of the origin of these neurotoxins. Okay, so when did the novel neurotoxins evolve? So what I'm going to tell you pretty much are two surprising stories. So we walked into this with various suspicions. We were studying this system because we thought we're going to get at a molecular novelty here. It's going to be fairly recent because the rattlesnakes alone are pretty young to North America, probably maybe 12, 15 million years or so. So we're going to catch a novelty essentially evolving. Okay. And here's some of the first experiments we did certainly made us think we're on the right track. So we compared the sets of genes present in non-neurotoxic rattlesnakes with those that were neurotoxic. Okay, here's an eastern diamondback. It doesn't have the neurotoxin genes. Here's a western diamondback. Here's an eastern diamondback. Neither of those have the neurotoxin genes. Mojave Rattler has a neurotoxin gene. So you think, there it is. So you want to know, where did these genes come from? Okay, so if we're looking and we're trying to think, well, which snakes should we sample? Phylogenetically, we're thinking, how do these various traits distribute across the evolutionary tree of rattlesnakes? And it turns out that neurotoxic rattlers are nested in a clade with a good number of non-neurotoxic snakes. And when you start thinking about polarizing the change, the most parsimonious explanation you come up with is not that the neurotoxins are something new in this clade, but the neurotoxins were around in the last common ancestor of all these snakes and have been lost independently multiple times. And before you suddenly jump and go, well, why would you lose a neurotoxin if it's and it is a very potent toxin, why would you not have it? Just hold that thought for a couple minutes. It's going to make more sense to you in a little bit. Didn't make sense to us at first, but this is, you got to deal with data. So we think that the neurotoxins are older and they've been lost in different lineages. And when we analyze these gene complexes, what we see is, what's happened is we infer that the last common ancestor of these rattlesnakes had a whole bunch of PLA2 genes, and that there's been various deletions within these complexes that have taken out 2 or more genes in various lineages. So the neurotoxic guy has lost a couple genes out here. The non-neurotoxic guys have lost stuff from the middle, and out go the neurotoxins. So the story is not a recent novelty, but a deep origin of this set of genes, including the neurotoxins, and then a recent loss. Okay, so if you think that's weird, it's going to get weirder. But that's, at least for me, one of the rewards of going and studying things that haven't been studied before, is you're going to perhaps find things that turn some of your thinking around. And here comes another example. And the other example is I've set this up by telling you that some species make hemorrhagic venom, some species make neurotoxic venom, but some species include individuals of each type. And this has been recorded in natural history and actually sometimes discovered for kind of the scary reason of somebody comes into the emergency room and they're showing symptoms that aren't expected from the snake that bit them. So there were 3 species we decided to study where this situation existed, where they make alternate venom types. And they do it, and there's different geographic patterns of distribution. So the southern Pacific rattler, shown here in yellow, out in southern California and into Baja, it is a predominantly hemorrhagic snake, but right there, that little blue dot, which you can barely see, which is right in near Riverside, California, just as a warning to you hikers, is a population that's neurotoxic. In blue here is the Mojave Rattler, stretching through, primarily through Arizona, bits of New Mexico, and into Mexico. It is a predominantly neurotoxic rattler, okay? But those little red dots show the location of populations that include hemorrhagic individuals, okay? And then the timber rattler, which is over a lot of the eastern United States, predominantly hemorrhagic, but all of these blue dots, which you may be able to see some of them, here we are in Oklahoma and parts of Louisiana and a little bit in Georgia, South Carolina, has neurotoxic subpopulations, or neurotoxic individuals have been recorded. Okay, so given that, those are entirely different genes, we were curious, how could we explain these alternative venom types? And the simplest thought we said, maybe this is a matter of gene regulation, that snakes have all the genes, but they express only a subset of them, or it could, of course, be polymorphism in gene content. So we analyzed the genomes of individuals of both types from all three of these species, and here's what we found. So again, these damn genetic maps, but here's the neurotoxic Mojave Rattler with his neurotoxin genes. Here's the non-neurotoxic Mojave Rattler from Arizona, and those genes are missing. So we're not talking about a pseudogene, we're not talking about SNPs here. Genes aren't there. Okay? Same species, presence, absence. Furthermore, this non-neurotoxic guy has 2 genes that are not present in the neurotoxic guy. And there's a gene in between here, it turns out to be a pseudogene that's present in the neurotoxic guy. So these things differ by the presence absence of 5 genes. You can find them in the same county. Okay, what's going on with Southern Pacific Rattler? Similar, but a little bit different than the Mojave Rattler. So there are the neurotoxin genes, and in the non-neurotoxic form, those 2 things are missing, and this one's missing. And there's no gene that's novel to this group that's not present in the neurotoxic guy. Okay, so how are we going to explain this sort of gene presence absence polymorphism? And here's the possible evolutionary mechanisms we were thinking about. You could have sort of the independent evolution of the non-neurohaplotype within each species. So maybe this is, you're deriving non-neurotoxic forms from a neurotoxic ancestor in each of these lineages. This could be a trans-specific polymorphism. In other words, they're evolving from a polymorphic ancestor and they're sorting out these haplotypes or maintaining polymorphism through speciation events, sort of the way the ABO system works in primates. Or, of course, and I'm sure it occurred to you, there could be hybridization between neurotoxic and non-neurotoxic species. All right, so we wanted to dig into this. But we didn't think the first explanation could really fit because there's no way to derive the non-neurotoxic haplotype from this because there's two novel genes down here that don't exist in the neurotoxic haplotype. So we're like, that doesn't make sense. So as we're sort of struggling to figure this out, Noah Dowell says, I want to go after the metalloprotease genes in these same species. And I'm like, Noah, that's a suicide run, okay? At that time, we had not studied any of the metalloprotease genes. And as it turned out, with these big gene complexes and half a megabase, et cetera, and you really have, because of the similarity of the genes, just people appreciate this, you really have to have perfect sequence data. So we had to do like back sequencing across all this. We couldn't rely on like Illumina sequencing to do any of this work. So I was like, no, this is brutal. But hey, it's his life, not mine. So he did it. And you may already be thinking about why would a snake, get rid of a perfectly good neurotoxin, et cetera. And if you're non-neurotoxic, how are you killing your prey? Okay, so we look at the metalloprotease genes in these same 2 individuals. So in the non-neurotoxic form, the one does not make the neurotoxin. He's got a complex of metalloprotease genes, and I'm just showing you like in bright red are the ones that are highly expressed in Venom, and these are also expressed in Venom, but a little bit lower levels. But when we look at this exact same gene complex in the neurotoxic individuals, the majority form in this species, it's got a 10+ gene deletion running right through the middle of the complex that takes out the genes that are essentially expressed in the venom. Holy smokes was the word I might have used. And first, you don't even believe these results when you get them at first because you're like, you're dealing with assembly issues and this could be an informatic problem or whatever, so you got to confirm it, multiple individuals and all this sort of stuff. No, this gene complex has a big deletion in it. And so this is telling us that the difference between neurotoxic and non-neurotoxic individuals in this one species found in the same county is the presence and absence of 16 genes. So there's 5 gene differences at the PLA2 complex, the phospholipase A2 complex, and 11 gene difference here. The deletion takes out 10 genes and part of an 11th. Okay, so this is a weird polymorphism. Gene presence, absence. I'll talk about that a little bit in a second. But it turns out that Noah's hunch was a clue to understanding the origin, at least in one case, of this polymorphism. Because when we then did the same work on this Southern Pacific rattlesnake population and looked at that neurotoxic versus its majority population, we discovered that the neurotoxic members of that population had exactly the same haplotype, that big deletion haplotype that we found in the Mojave Rattler, right to the base of the edges of that's all this, what this data shows is basically right down the borders of the deletion of the exact same nucleotide. Okay, so clearly there's a shared haplotype between the two populations, and that eliminates the independent evolution scenarios, but it still leaves in play the possibility that this is a polymorphism, that each species is inherited from a common ancestor, or it could be hybridization between the neurotoxic Mojave Rattler and the non-neurotoxic Southern Pacific population. So we did the Abba-Baba test to look for introgression, and a whopping signal. Quite clearly there's been hybridization. So it's satisfying, and of course there's a lot of interest in hybridization, Molly, between species. And fortunately the press didn't catch this up, that snakes are swapping toxin genes out there. I'd like to write that headline. Anyway, so we have at least one case of hybridization. We don't understand the origin of the other polymorphisms yet, but there is hybridization going on, and that hybridization can mean the exchange of different types of venom genes between different populations. Now, I just want to make a couple comments because we hadn't seen anything like this. These really extremely divergent haplotypes, these huge structural variations, presence, absence of several or up to 10 genes between different species. If you can think of any examples in animals or even in plants or microbes, please share them with me. I'm sort of collecting them. But I gave this talk somewhere in London, and somebody pointed out that natural killer cell receptors on primates, including us, show this type of variation, this kind of geographic variation where presence, absence of genes, multiple genes in large haplotypes. And that made us start thinking that maybe, first of all, you may want to mention these complexes are unlinked. So there's no way to coordinate these deletions in any sort of way. They're not linked genetically. They're on different chromosomes. But so we think what's going on, by analogy to the natural killer cell story, is that this could be an arms race. Those genes are thought to be in an arms race with parasites, with pathogens, human and primate pathogens. In this case, we know these snakes, they prey on different kinds of animals. We know that numerous prey have evolved resistance mechanisms. We also know that these snakes are preyed upon by different predators. So there's some complex interaction going on out there in the real world that may be maintaining the polymorphism in certain snakes, and other snakes have lost this polymorphism, at least until they regain it by mating with another species at some point. So that's what we've learned in a nutshell so far. As a geneticist, I thought it was an interesting sort of form of genetic variation to find. But as a developmental biologist and sort of evo-devo guy, I was really concerned that as much as you can tell, I like snakes, and there's many, many more stories to pursue. Having had the benefit of studying fruit flies for so long, you wish you could do more things experimentally. And again, same talk I gave in London. At the end of the talk, this Dutch fellow named Hans Cleavers comes up to me after the talk and he says, I've got two guys in my lab. They grew venom gland organoids from cobras. Hans is a fantastic stem cell biologist and done a lot of things that are medically relevant. And I said, Hans, what the hell are you got people growing cobra venom glands in your for? Well, they wanted to. Good enough reason for me. So it turns out, and we've now replicated this in our lab, that you can, under the right conditions, grow these organoids from primary venom glands, and they're passable in the lab. And so we're hoping that we can treat this system as we would anything else in developmental biology, that now venom gland cells and gene regulation and all that is open to us experimentally, reporter genes and transcription factor biology, et cetera, is open to us because you can grow these glands in culture. And it never would have happened if I hadn't just opened my mouth one day in London, and Hans didn't have a couple of crazy graduate students doing something that was so far off the map of what the lab was doing that we would have never imagined that you could do this kind of thing. Thanks for listening. Are there young learners dying to get in here at the moment, or I don't know what we've got for time for questions.
Yeah, we have time for a few questions. Yeah.
I have a question about snakes. So, if you're looking to other groups of venomous snakes,
Yeah.
Do you see expansion of these gene families or expansion of other gene families?
Right. So cobras, for example, have expanded other gene families in making neurotoxins. And yeah, so I mean, there's phylogenetic sense to this in terms of what got expanded. But cobras have gone a different road in making neurotoxins than rattlesnakes and old world crotalids have. Yeah.
And also, I remember the family of king snakes contain a lot of species, but only a small portion of them are venomous. So is it because they kind of use a different mechanism to evolve that?
You said king snakes?
The family of king snakes.
King snakes. So if you're talking about the constrictors found here in North America. So I want to make sure.
I only remember some species of that family.
Okay. So king snakes are a really interesting case, and I'll give away the next wave. It's a little different story. King snakes are named king snakes because they are cannibals that will eat snakes. And they take down rattlesnakes with impunity, eat them whole. Go on the web if you want to see it. There's plenty of YouTube videos of king snakes killing rattlesnakes if you like that kind of blood sport. So king snakes are harmless constrictors. And in many parts of the United States, they are preying on poisonous snakes. And we've known since 1950 that they're resistant, but the mechanism of that resistance is not known, but I hope to come back to Michigan before, I don't know, too long, and tell you, because we're working on it. But so it's not, the king snakes aren't venomous, they are resistant to the rattlesnake venom.
I think you might mean like other colubrians, like Savannah twig snakes or boomslimes in that general clade. Talking about the family.
Okay, because king snakes is Lamprepeltis here in the United States. So that's why I just want to know which group because maybe kings have names somewhere else in the world. There's not a lot of formal literature. So you're saying about things like boomslangs and stuff that are.
No, I think you were talking about. You're just asking about other snakes in the same family, king snakes that are venomous. So there are a few.
Oh, you're talking about in terms of colubrids in general, like rear fang snakes and things like that. Yeah. So yes, so there are members of colubrids that are rear fanged and venomous. And since you know about these things, there's always herp fans in every audience that know more than I do, and I'm about to be in trouble. So, but I just don't know the toxin. I don't know if we know the genomics of the toxins yet, of where they came from. But they're going to be different than these guys. To my knowledge, the recruitment of this subfamily of PLA2s is unique to crotalids, and other neurotoxins have come from other places, and certainly elapids, cobras, and the Australian snakes have come from other gene families.
Okay, if anyone has time for, no, okay, no more questions. Sorry.
Hey Keith, how you doing? All right, see ya.